Okay, so this is the last section of the class. We're going to talk about circulatory tumor cells that are also known as CTCs. So let's start talking what is a CTC. So a CTC stands for, here you can see it, the C is for circulating tumor cell. Yeah, and so these circulating tumor cells, and you can see here in the graph, they have the characteristics that they go for a primary tumor. So primary tumor here in this region, and from this primary tumor, it goes into the blood. Yeah, and it starts combining with all the blood cells. Here in this schematic, they try to make it simpler for you, but the proportion of blood cells to CTCs is very, very, very small. So it's very difficult to study them because if you have thousands and thousands of cells and you have only few of them, few of the CTCs, to find them and to follow them is very difficult. So here you can see the CTC traveling through the blood and many of them will die. However, few of them can go outside and attach and produce metastases. So remember, important thing about the CTCs is that they are cancer cells that detach from a primary tumor and they go into the blood. And they are important. Why? Because they can be important for the formation of a new tumor. Again, this is very unlikely that happen. Many of them will die. Most of them will die. And here they show it like as a single cell. They can go. They can find individually or they can find in cluster of cells. Yeah, these are more frequent, the single cells CTC, while the clusters are found less frequent. However, the clusters they have been found to be more likely to invade other tissues. So understanding and detecting these structures, these CTCs, is very important. And not only so, blood samples can help us to see how many CTCs, and I think all of us we can have some degree of circular tumor cells or cells that are benign that go there. However, when it goes beyond certain number, it could be a good biomarker to study cancer. However, to study this in the blood vessels of the patient is impossible. Even if we tried, and like right now, the techniques, it will be very difficult to study that in vivo, even in vivo in animals, is just very difficult. So what I'm going to show you is what we try to do, yeah, using fruit flies to try to understand this question. So previously in the lab that I was working, they were trying to study this in vivo, yeah, that again there was a lot of studies in ex vivo, but with Drosophila we have so many genetic tools that they said maybe we can do it. And again they use RAS and a tumor suppressor, modification of a junction, genetic perturbation in a junction, and that produced a highly invasive tumor. So here you have the tumor that is going to move from the brain to nerve cord of the flies, ventral nerve cord. So you create here the genetic perturbation and it will start slowly invading. However, what they found is that in addition to the invasion, they start finding all these dots, all these dots that are everywhere, that they start migrating from one region to another one. And the question that they had is, are those what are those cells? Are those pieces of tumor that are moving from one place to another one? Are maybe immune cells that So they are able to move. They are very small and they are moving from another region to another region. And this work was never published in the previous lab that I was. It was just very interesting and it was an effort. But as it was, they were working here in the larvae. They were only imaging few minutes, yeah, and and it was the larvae are constantly moving, so they try to cool down the temperature of the larvae so they stay still for a longer time. And but if you do it for a long time, they end up dying. Okay, and the larvae, in my experience, has been if you freeze, reduce the temperature of the larvae in the long term, they will die. So you end up a little bit creating a small problem in the experiment. So here you can see all those things that they thought maybe this could be CTCs, pieces of tumor that go to another place. And what they were trying to think is, could we have individual migration that it goes in attach in the other part? And this could be a good way to study that. And what they were not able to see if they were able to reattach and help to the invasion process. Yeah. So what they were trying to do, they generally took pictures of the beginning and at the end of the invasion, but they didn't have all this process. Yeah. So they kill one larvae and they take a picture, and they kill the other larvae and they take a picture, and they find that the end process was the same. However, they didn't know what happened in the intermediate stages. And what they found, it was this, but it was not a strong evidence. And they decided to quit this project because, of course, it was very difficult to work with larvae. Okay, so now I'm going to show you a comic. So if you end up to go to grad school, you will hear about this, especially in biology. And this person became famous for he was doing a PhD and got tired, but turned all his frustration related to finishing his PhD into a business. And now he has made a lot of money, and they just tell the jokes of all PhDs that they happen. So, for example, if you had problems with any of my courses with research project, welcome to the club, all of us. And if you are interested in going into grad school, uh, this is something that is very common, and you need to learn to in all areas you will find problems that you will eventually solve. In research, you can spend 5 years and say this is as far as we go, and we were not able to completely solve the problem. We just understand it a little bit better. Okay, so now I want that you go here and give a look to this paper that says CTC micropublication, and give it a read and try to look for the bow tie. Yeah, try to look how we went in this paper and went from the more general to the most again to the results, and then to the implication of this work, and try to be aware of that. Look at the figure and try to give a quick 5 minutes and try to see how those papers are written and the main idea about CTCs. So now that you read the paper, I tell you that we try to study how tumors that are pieces of tumors that can be attached during the like development. So this is a movie from a different lab, and here is a link. And what they did is something that I thought that it was impossible. That is to image this very early stage of development. Here you see each one of these is a leg here. You see the legs here. Here is the wing of a fly that is developing, and as you see, it suffered a lot of changes in a very short time. And as a result of those short-term, very fast changes, what is going to happen is that there will be a lot of mechanical pressure, mechanical tension in the tissues. That if you see those tissues don't still remain uh, healthy. But I wanted that you think what happened if you have all that movement and you have pieces of tumor there. Yeah. So under all these mechanical traits, so on a typical growth could stand all that mechanical pressure, all that mechanical pressure. So we thought maybe this could be a good way to study CTCs because the tumors will end up breaking, and there will be many things that it could be happening. Okay, so now we are going to study the cephalic complex, and here you will see the different components. Here you find the legs, as I told you last time, the wings. That you don't see, there should be another wing here, but this image doesn't allow us to go and see the other region because the angle of the in which the pupae is located. If we turn it a little bit, we will be able to see this wing, and we will lose this one. Here there will be a boundary located in the distal part. And what we did is create genetically, using different tools, different type of tumors. Similar to the previous experiments, we use RAS-B12 and we use another tumor suppressor, a mutation in a tumor suppressor. So what we saw it was that the tumors start separating and start moving in random way, but they show some patterns when we see them when we analyze all of them in the same context. So we establish a specific temperature to activate the tumor at a specific time. Yeah, this is something that is good about genetics with fruit flies. So if we express the tumor suppresses from the beginning, the genetic perturbation, the RAS-B12, and the mutation in the tumor suppressor, the fly is going to die very early in development. However, if we keep those genes expressed at a low level and we express it at a specific time, we increase the temperature, they will those genes will start being active and producing the tumors at a specific time, and this will allow us to allow the pupae to survive, and also to study these specific conditions in the pupal stage. And again, this is the genetic perturbation that we use. So tumor suppressor, a scribble, and if you take genetics, we will talk about these flipases that are genetic tools that allow us to produce clones. Yeah, specific. Instead of being the tumors everywhere, you can put patches of cells with these specific genetic expression. Some of them, this one is random with the Hitchcock flipase, and this one is only in the eyes, and they can travel and go into the legs. Okay, so the idea is that we use a tool, genetic tool, that allow us to make patches of tumors, yeah, where the tumors are expressed in a specific region. And why this is important? Because this allow us to study how these tumors can travel. Okay, so this allows us to study. We found that the tumors are always travel, and they detach and travel to different regions. Okay, so let me show you. So this was in an early stage. We have GFP to label things. Yeah, I try to simplify it with the schematics, and I need that you have faith. Yeah, what I'm going to show you, in the sense that as you're not an expert, you will see Nicholas is just green spots moving. Yeah, so I'm going to show you movies that they have that are not color coded completely, and later I will go into each one of the movies and color code it for you. Yeah? So here you will see, for example, this is the normal data, yeah, the raw data, and here you see a dot that I will magnify that figure later. Here the arrow, and I will put an arrow that it will show you where the tumor is moving, and here equal when they are not moving. And here you see a raw data that is And, that is how you will see without the label. So as you can see, there are dots that are moving. Some of them are single, some of them are in groups, and you can see it in both. Here you cannot see clearly the legs. I haven't labeled it for you. Here what we are only labeled is the tumors with the GFP. And the green region here is the pupil case that is autofluorescent. Okay, so key thing, circles that are moving everywhere, and those pieces are pieces of tumor. We went and started doing multiple movies, and in each one of those we start finding, okay, it seems that in all of them we find this tiny tumor. So the problem that we had before, here we didn't find it. We find that always we find these tiny tumors traveling. Here you see another example of a tiny tumor detaching and moving between the legs of the flies. And here we find also the same. So don't worry, I know that is overwhelming. There is a lot of things. What I wanted to show you is that it's different from the previous behavior. This behavior happened in every single movie that we study. And every time that I'm not going to show you all the movies. I haven't the time to analyze all of them that we have captured. We took 3 for the paper that to analyze, that they were the easier ones, and that I already removed the background. You know, this pupil case, I had to remove them so to make it simpler. For the analysis, there are more that I haven't done analyze more, but I have seen more CTCs. In I think in every movie that I have seen these kind of things. Okay, so now that you know how they look, now I'm going to show you what we did. So here you see large tumors, yeah, and you will see these dots, yeah. And what we did is exactly this. We played the movie, and here what we did is to start following the trajectory of each one of these CTCs. So in yellow you will find, sorry, in white you find the first time point, then second time point in red, then orange, sorry, this is pink, and then yellow. And here we did something similar. So this allows us to see which is the trajectory of the CTCs and analyze the velocity of the CTCs. If you took cell biology, I, I try to show you that this is not complicated to do, that with ImageJ we can do it, and actually this is something that we have the data already, and if you want to join one of these projects, we need to, we haven't finished that project, but we we measure the speed of those CTCs and the trajectory, and we just have to analyze it. Okay, and here, as I said to you, white is the trajectory, and time 1, time 2, time 3, time 4, and we can even go further and keep analyzing that. So this is one thing that we did. We saw all those CTCs and we start trying to see the general patterns, and we start quantifying how many there were. We start trying to get all the cell parameters related to the CTCs, because as I told you, in vivo they are very difficult to study. Generally, what you do is study them in vitro. You take a blood sample and you put it in a petri dish and you see if they are alive or not, or you can transfer some of those CTCs into mice and try to see if the mice can get cancer, or these type of things. So here you see a trajectory. Now I'm going to show you very weird things that we found there, unexpected things. So here you see a CTC, and here I'm going to zoom in this CTC. So here what you see is 2 pieces of leg in blue that I labeled there that are around the CTC. Yeah, and if you see, those 2 pieces of legs are getting very close to this very tiny CTC formed by multiple cells. So as you expected, it's like an elevator is closing and you have your body or your head there. So you know that you have to move away from there. So look, I'm going to show you when we have in 3 dimensions. what happened to that CTC? Well, the legs, I told you there was a lot of mechanical pressure, that the legs were moving, that there was a lot of force. So what happened with that CTC is that they are going to get squished and they are going to break into many tiny cells. Yeah. Okay, so now I'm going to show you what happened on the top, the middle, and in the bottom of the CTC. The microscope allows us to observe each one of the each one of these sections. And just I don't put it all at the same time because it will look like too many things happening at the same time. So you will see something like this. Yeah, so let me show you here on the top. You see that it got a squish. Here in the middle, it got a squish in the middle in this area, and here in this area of the bottom, it got a squish. So in other words, what we were showing is that CTCs are interacting with other tissues. Yeah, they are able to move. We can follow the trajectory, and there are a lot of things that depend on the microenvironment. Yeah, in this case, you have legs that are moving. They can break into pieces, or they can fuse with other ones. So we were trying to see if we see a random behavior or we find some predictability on the type of movement. Yeah. So this is something that we already found, that we have the data, and hopefully I find somebody that helped me to finish this paper. So we found that there was so many different trajectories. However, when we analyze all these different trajectories, we see that all of them follow the same path. They move from the top, yeah, and to the bottom, yeah. And there was some of them that go into this direction, but they never went into this direction, yeah, yeah. They always go down, and then they can start moving in this direction, yeah. And some of them they can go back here and here, yeah. So it was very interesting because it seems that they are moving passively, that the hemolymph, that is the liquid that in which the blood of the flies, the liquid in which this imaginal disc, the legs are floating, yeah, is the one that is in charge of the movement of these pieces of tumor. So we find that when we analyze it, that there was predictable movement, and we find that the cellular environment seems to be very important for these CTCs. So instead of having their own movement, they are moved by the current. So if you remember the currents of the ocean, it looked like that they were moving around the legs. They were trying to escape to not get squished, and more than having an active movement, what I mean, like I will show you that some of them, if the legs close and form a barrier, they will get stuck, and some of them will just moving, trying to avoid the legs. So again, I show you that there was a space, and when there is a space, they can cross this region. Yeah. However, as the leg develop and start becoming thinner and thinner, they elongate. So they are going to start touching the legs. Yeah, there is less space because the legs are developing. So what is going to happen? I'm going to show you an example in which sometimes when the legs start growing too fast, the tumors start getting stuck there. Okay, the pieces of tumors, and they form a blockage. So here you see that the tumors just got stuck, and they start. We don't know if they will fuse completely or not, but they start getting stuck in that region, and they start interacting between each other. Yeah. So again, same idea. Microenvironment seems to be very important to try to understand what exactly is happening there. Yeah, the CTCs seems to, instead of having individual movement, they seems to be start following a current and depend on the tissue around it. That is something that we found fascinating. However, despite all this analysis, yeah, we found that although we found that it's easy to see pieces of tumor detaching and traveling everywhere, and we find we found that, and we know that this has happened because the molecules have also GFP, so only the ones that have GFP are there. So it allows us to see that they used to belong to a tumor, yeah, and we see some of them that they seem to potentially fuse, and some of them that they seems to attach into a tumor. We didn't see any tumor growing. Yeah, why? Because although this technique is really good and you can analyze in the larvae, we can only analyze few minutes and or few hours if you sleep it and bring them back to a normal temperature and try to look for the same spot. Here you have around 6 hours of imaging, what is much longer than in other samples. However, after that, there is a process called the head aversion. So all the body move a lot, and later when we try to go and study the CTCs, they are gone. They are in other places. We don't know exactly if they go inside of the body of the fly, or they are destroyed, or we don't know exactly what happened. Or the pupae is, if we create more CTCs, we create stronger perturbations. The pupae just died. Yeah. So many times you say like, okay, we made this strong perturbation. Now when there will be this big movement, we will be able to see where the CTCs are going to go on and follow them for a longer time and see if we can see this process of colonization. However, many times the pupae just died at that time. So this is again things that we need to do future work to try to understand this better, to find if we can do more specific perturbations in a specific time that affect our tissues and don't end up killing the pupae. And in that way, I think this could be a good way to study that. Again, the mechanical forces play a key role during tumor development. So I show you that during the process of epithelium crowding. Here I show you that again, but just showing you that as the tumors grow, they are not as stable as other tissues. So while you don't see a lot of pieces of normal tissue dying in big pieces, although there are some that definitely go there. When you have a tumor, it's more likely that they break. So it's something that is interesting, and see them moving is also very interesting. Okay, so to summarize all the things that we talked today, what I wanted to show you is that when you go and study tumors, yeah, I show you different behaviors, yeah, that are happening. So I show you a lot of images, and I don't want that you memorize all the images. What I want is that you remember that although when you study from the beginning to the end, the process seems to be tiny tumors. Then at the end, you see, oh, there was big tumors and they move everywhere and they kill the pupae. However, the way in which they do it, when you go and do imaging analysis, is that there is a lot of variation in the way that the things could happen. And it's fascinating to try to understand that, but at the same time, it makes it more difficult to analyze. Hopefully one of you want to join this type of projects, and we can work together and try to have, again, similar to the micro publication that you read, another micro publication with your name. Okay, if you have any doubt or any question, let me know. And with this, we finish the topic about the CTCs. Have a good day, and if you have any questions, please do not hesitate to email me.